really unknown. This unexplored territory, that's really what is appealing to me. So I like it when I have no idea how things work, and then I can come up with models how that could actually really play out. Most of our studies concern CD47, which is a membrane protein, and so it basically is on the cell surface of every cell. And it basically functions as a don't eat me signal. So immune cells are watching out for viruses and bacteria to see what's foreign. And so people thought that this is the only function of CD47, and it's on the cell surface. But when we looked more closely, we found that CD47, a lot of it is actually inside the cell. And this was very puzzling. And then we actually found out that the intracellular CD47 has its own function. And it actually has the opposite function of the surface molecule. And those different locations then lead to different functions of this one protein. Every cell expresses CD47, but the cancer cells express it at very, very high levels. And if we only give a little bit of the drug, usually the cells that expresses the most would be targeted first. And then normal cells, they express very little of the surface CD47, and they actually should not be targeted. Cancer cells actually want to survive, and to do so, they want to get rid of the intracellular CD47. And what we found out is the surface CD47 is produced by a long mRNA, and the intracellular CD47 is produced by a short mRNA. So if we target this long mRNA, we basically can get rid of the surface CD47, which is actually the oncogenic form, but still keep the tumor-suppressor CD47 isoform. This is actually very new because so far you usually always target the whole protein, and we can now target a specific function of a protein, and we get rid of it, and then we should actually have a more specific therapy with less side effects. We basically set the stage for a new therapy, and so currently we are really focusing on CD47 because we want to understand CD47 better, and then maybe we can extrapolate what we learned from CD47 to other proteins and also learn how these longer and shorter mRNAs can then lead to differences in function. And I think that's really exciting.